Welcome back friends. Na leo tuangalie kuhusu enzyme cofactors. Kwenye kipindi kilichopita tuliangalia kuhusu allosteric inhibitors na kuhusu enzyme inhibitors. Kwa hiyo don't forget to subscribe to the channel and to share the link to your fellow students wakati tunaendelea na vipindi vya biology, chemistry, physics na mathematics kwa level na A level. Now, let's continue with enzyme cofactors. A cofactor is a non-protein substance which is essential for some enzyme to function efficiently. Or sometimes, an enzyme cofactor is a non-protein helper needed by many enzymes for catalytic activity. Kwa hiyo cofactor nini? Cofactor yenyewe huwa ni non-protein, sio protein, kwa sababu enzymes they are protein in nature. Now cofactor they are non-protein, lakini zinasaidia enzyme kufanya kazi. Kwa hiyo in absence of cofactor, enzymes zinaweza zisifanye kazi kabisa au zikafanya kazi bila kuwa na efficiency kubwa. Lakini cofactor kiwepo inakuwa ni kama helper, inaisaidia enzyme kufanya kazi vizuri. Kwa hiyo unlike enzymes, cofactor are stable at relatively high temperature. So cofactor, because they are non-protein, has you affected na nini? Na temperature. Cofactors may vary from simple inorganic ions to complex organic molecules, and they may either remain unchanged at the end of the reaction or be regenerated by the later process. Kwa hiyo cofactors zinaweza zikafanya reaction na zikabakia vile vile, au sometimes cofactor unaweza kukuta ile reaction ina ivunja cofactor, lakini no problem because a cofactor is the helper. It is involved in the reaction as the helper. Na most of times unaweza kukuta kwamba body inaweza kapata tena cofactor nyingine kwa njia ambayo ni very easy. Now cofactor may be bound tightly to the active site as a permanent resident or may bind loosely and irreversibly along with the substrate. So we shall see later, baadhi ya cofactors zina bind permanently na nyingine zina bind kwa muda tu. Tuna three types of cofactors. Ya kwanza ni inorganic ions, na they are normally called as enzyme activators. Inorganic ions are normally called as enzyme activators. Then we have coenzymes. The coenzymes, then we have protetic groups. So starting with activators or inorganic ions, these are substances which are necessary for the function of certain enzymes. They are believed to form a bridge between the enzyme and the substrate, thus binding them together. Or the activator assists in forming the enzyme substrate complex by modeling either the enzyme or substrate molecule into more suitable shape. By doing so, they increase the rate of reaction catalyzed by that enzyme. Kwa hiyo cofactor zinasadikika kufanya bridge kati ya active site ya enzyme pamoja na nani na substrate. Kwa activation are usually ions. Activators are usually ions of metal atom such as magnesium, potassium, calcium, iron, can be iron 2 or iron 3, copper and zinc. Now we have some of the examples here. The first example, the enzyme thrombokinase. Enzyme thrombokinase, which is essential in converting the soluble fibrinogen monomers to insoluble fibrin during blood clotting, is activated by calcium ions. And ukienda kusoma mbele kuhusu mambo ya blood clotting, utakuja kuona kwamba kuna vitu na vita blood clotting factors, or sometimes they are called von Willebrand factors. Now, one among the factors which are involved in the reactions or in the pathway of blood clotting is calcium. Now, calcium being the factor is the activator of the enzyme, which is thrombokinase, catalyzing the process of conversion of fibrinogen monomers into insoluble fibrin. And the second example for activator is salivary amylase, require the presence of chlorine, presence of chloride ions before it will effectively convert starch to maltose. and the presence of chloride ions in the mouth is very easier to get chloride ions because we normally consume salt which contains sodium chloride. Now let's jump to another type of cofactor, which is coenzymes. Coenzymes, they are non-protein organic substances or prothetic groups which are essential to the effective functioning of some enzymes, but are not themselves permanently attached to the enzymes. Now we shall see later about prothetic groups, and if you are serious with me, you may sometimes wonder the difference between coenzymes and prothetic groups. The major difference between coenzymes and prothetic groups. Coenzymes, they do not bind permanently, or they do not attach permanently to the enzymes. But prothetic group, they bind permanently. Now coenzymes are organic molecules. They are organic molecules cofactors. So most vitamins are coenzymes. All raw material from coenzymes are made. Kwa vitamini nyingi ni coenzymes zenyewe au zinatumika kutengenezea coenzymes. All coenzymes are derived from vitamins. Example ni NADP, coenzyme A pamoja na ATP. Kwa hiyo hizi tutaenda kuzidiscuss baadhi kwenye kufanya kazi, kwamba kwenye functioning mode ya coenzyme, a coenzyme assists the enzyme in transforming the substrate by act as coenzyme. First inaweza ka act as an electrons or hydrogen atom being removed from the substrate. Can act as an acceptor of electrons or hydrogen atom being removed from the substrate. Lakini pia inaweza act as a donor of electron or hydrogen atoms by contributing electrons or hydrogen atoms in the synthetic reactions. Kwa hiyo inaweza act kama donor au kama acceptor wa hydrogen au electrons. Kwa kuact kama donor au acceptor wa hydrogen ions, at the end of the day zinaweza kusababisha reaction yako iweze kuproceed kama ilivyotakiwa. So coenzymes in general have been found to associate enzymes concerned with oxidation reduction reaction, kwa sababu mambo ya addition na removal of electrons and hydrogen, hizo ni oxidation reduction reactions. Now let's discuss the important coenzymes in cellular metabolism. Our first coenzyme is NAD. NAD is nicotinamide adenine dinucleotide. Now, this acts as a coenzyme to dehydrogenases. Dehydrogenases, they are enzymes which tend to remove the hydrogen from certain molecules. Now, NAD tend to accept that hydrogen and transfer it to other reactions. Now, as you can see in this example, you have this molecule with a hydrogen molecule, and then here the molecule is idle because NAD has taken the hydrogen molecule. Now, NAD is derived from vitamin nicotinic acid or niacin. So, NAD is derived from niacin and can assist in both a reduced and oxidized form. It is oxidized state, it function as a hydrogen acceptor. Kama ambavyo unaweza kuona hapa kwamba huyu hapa ana hydrogen, ametransfer hydrogen kwa NAD, huku NAD ana hydrogen, huyu hapa ni oxidized substrate. Kwa hiyo ni hydrogen donor. Lakini the reduced state of NAD can act as a hydrogen donor. Huyu anakuwa hydrogen donor, donate hydrogen kutoka kwenye B catalyzed by enzyme 2. Kwa hiyo ndio hapa NAD ana act kama cofactor hapa, NAD ana act kama cofactor hapa. Kwa hiyo kwenye mazingira hayo unakuta kwamba where E1 and E2 are different dehydrogenase enzyme, Net effect is two hydrogen transferred from A to B. Here the coenzyme acts as a link between two different enzyme system E1 and E2. Kwa nakuta nadi ameact kama link kati ya reaction ya huku na ya huku. Huku amechukua hydrogen, huku amempa ile pale. Lakini kama nadi asingekuwa, maana hizo reactions zingeshindwa ku take place. Nadi ameact kama ni cofactor hizo enzyme control reaction zote mbili hapo. Lakini our second example of cofactor is NADP. NADP is nicotinamide adenine dinucleotide phosphate. Hii inatumika sana sana kwenye respiration, lakini pia kwenye photosynthesis. NADP acts as coenzyme to hydrogenesis in respiration process. Both NAD and NADP are derivatives of the vitamin B. Vitamin B, which is called niacin. Now the coenzyme accepts hydrogen from the substrate to become reduced. Later pass the hydrogen to another hydrogen acceptor, so becoming oxidized and lead to accept other hydrogen atoms with the dehydrogenation enzyme, as have been illustrated in the diagram above, which we have already explained. Another important cofactor is coenzyme A. Another important coenzyme is coenzyme A. This is another coenzyme which contains a derivative of pathogenic acid B complex vitamin. Now, this is not involved in transferring hydrogen atoms or electrons, but is the carrier of certain useful fragments of organic molecules such as acetyl group. Kwa hiyo kwenye process kama Krebs cycle tenda kuenda kwenye respiration, acetyl coenzyme A, coenzyme A ana carry acetyl group from outside of mitochondria into inside of the mitochondria, yani into the matrix of the mitochondria. So in the process of respiration, coenzyme A acted the carrier of the acetyl group from outside of mitochondria into the inside of mitochondria. It acts as conjugation with enzyme involved in various biochemical reactions, notably the oxidation of pyruvate, that is pyruvic acid, via Krebs cycle. So as you can see this diagram, but sometimes you cannot understand this diagram if you have not studied the topic of respiration, but we shall see later. Pyruvate is the end product of glycolysis. Now pyruvate from glycolysis want to enter the Krebs cycle, which is these reactions, series of reactions. Before entering the Krebs cycle, first it should undergo oxidation, which is the removal of hydrogen ions and decarboxylation. Pyruvate is the 3-carbon molecule. So removal of a single carbon dioxide molecule and removal of hydrogen makes it to be acetyl. Now acetyl is the 2-carbon molecule, and acetyl should be taken into the Krebs cycle. Now to enter the Krebs cycle, acetyl should be taken by the coenzyme A. Coenzyme A anamchukua acetyl kutengeneza acetyl coenzyme A, then anampeleka kwenye Krebs cycle kwenda kumaliza reaction. Na kwenye kipande hichi group ya mwisho ya cofactor ni prothetic groups. Now tunachokisema ni kwamba like coenzyme, prothetic groups are organic molecule, but unlike coenzymes, they are bound to the enzyme itself. Most enzymes are made up of protein chains only. Many other enzymes contain an additional non-protein portion, that is prothetic group or conjugate protein. So enzyme is equal to protein plus non-protein material, that is prothetic group or conjugate protein. Now the major difference between prothetic group and coenzymes is that coenzymes, they do not bind to the enzymes, while prothetic group, they bind to the enzymes permanently. Now the first part, which is protein portion, composed of only chain of amino acids, called the apoenzyme, and the apoenzyme and the prothetic group are collectively designated haloenzymes. So apoenzymes plus prosthetic groups, they are called haloenzyme. Now enzyme is equal to protein plus prosthetic group, or enzyme is equal to apoenzyme plus prosthetic group, and the whole molecule is called haloenzymes. So example prosthetic groups, we have FAD. FAD is concerned with the cell oxidation pathway and the part of the respiration chain in the respiration process. So the function of FAD is seen as if it is similar to that of NAD. But for example, if this reaction could be catalyzed by NAD, it could require two different enzymes, E1 and E2. But because the reaction is catalyzed by the enzyme and the cofactor which is involved is FAD, which is prosthetic group. Now, FAD has already bound to the enzyme. Only a single enzyme catalyzes the process of transfer of hydrogen from A to B. But there in NAD, tungeona enzyme mbili zinahusika. Kwa hiyo ni difference. FAD inakuwa tayari imeshabind kwenye enzyme na sio ina bind wakati wa reaction tu. Lakini pia tuna heme. Heme nayo ni prosthetic group. Heme is an ion containing prosthetic group. It has the shape of the flat ring, polypeptide ring as found in chlorophyll with an ion atom at its center. Example of heme protein include hemoglobin, myoglobin and the cytochromes. Hizo zote zinakuwa zina iron. Lakini kidogo tuguse hapo function of polypeptide ring kwenye heme group. Kwenye heme group kuna polypeptide ring. Function zake nini? Ya kwanza, ina act as electron carrier. In accepting electrons, the ion is reduced. Kwa inatoka kwenye ion 3 kwenda kwenye ion 2. In handing on electron, it is oxidized to ion 3. So cytochromes act as electron carrier in photosynthesis and respiration. So the ion can accept electron or can release electron, and by accepting or releasing electron it becomes oxidized or reduced, and the cytochrome they act as electron carrier. Cytochrome is a protein pigment containing iron that is capable of being alternatively oxidized and reduced in mitochondria and chloroplasts. Lakini pia polypeptide ring inaweza kaact kama oxygen carrier kwenye hemoglobin na kwenye myoglobin in vertebrates, giving the blood a red color, have high affinity to oxygen. They are oxygen carrier protein. Now hemoglobin has 4 heme groups. One of them is drawn below. So this is an example of the heme group in the hemoglobin, and as you can see, this is one a heme molecule, one of the heme molecule containing iron. Hemoglobin has 4 heme groups. That's why one hemoglobin molecule can carry 4 oxygen molecules consecutively at the same time. In the next video, we'll be discussing about water. So in this video, let's end up here. And this is the end of enzymes. In the next video, we'll jump to another part of biochemistry, which is water, and we shall discuss functions of water and all the facts about water. Don't forget to subscribe to the channel and to share the video to your fellows so that they get these topics. Thank you, everybody. Have nice studies.